We're now going to get into the process of cell respiration, and right here is an overview of the entire process. We're definitely not going to talk about it all in one clip, but I want to show you where we're going and where we're starting. So what we're doing first is this idea, it's a three-part process. That's kind of how we break it down. And if you think that this is where we left off in the last chapter, was the production of glucose. So glucose is now going to be what is called oxidized. We're going to lose a lot of electrons throughout this process. We're going to convert a lot of this energy that this glucose stores into ATP molecules, where ATP is more the molecule that cells are going to use to do things. We've talked about pumps in the cell, so ATP can be provided for those. And there's other processes in your body that would require this ATP. So this is the first step. It's called glycolysis. And if you see glyco, that kind of is a prefix if you've heard of glycogen before, with glycogen being the polysaccharide of an animal cell. And then lysis is to break. So glycolysis is really the breakdown of glucose, really the oxidation of glucose. So this process, you recognize some of these words from even photosynthesis. Glucose is one of them. PGAL is one that you have recognized. This is a new word, pyruvate. But that is the process of glycolysis, this top box right here. And so we'll talk about that and make sure we understand it. From there, the cell, depending on if the organism is going to go through anaerobic respiration or aerobic respiration. Aerobic respiration would require that there's oxygen present, whereas anaerobic is going to be that the organism chooses or does the process that doesn't require oxygen, or there is no oxygen available and this organism lives there, so therefore they go through the process of anaerobic respiration. We will go this direction first in our concept, and then we'll retreat over here and say, what if we went the other direction? All right. From there, this process is somewhat broken down. We're going to try and talk about what happens when we make this molecule of pyruvate before we get to this cycle, which is called the citric acid cycle. So we'll kind of break this down into its own little kind of box. That becomes its own little cycle in the process. This becomes its own process that will really isolate. And then finally, the electron transport chain. So some of this stuff will be review or concepts that you've heard of before. So therefore, it's not going to be that difficult to grasp. So let's proceed into the process. So the first thing we have to understand is where things are taking place. So glycolysis, which is this first bracket here, takes place in the cytoplasm of a cell. So they don't even need the actual mitochondria, which is the organelle that really you associate with this process of cell respiration. Glycolysis takes place out here, and we'll talk about the consequence of that later on. From here, the next two, well, from the remainder of this is going to happen in the mitochondria itself. And we'll dissect the mitochondria when we get there to talk about more specifics as to where it actually occurs. So let's dive into glycolysis. So here's a breakdown. The orange circles represent carbon atoms within the molecule. The yellow circles with the P inside of it represent phosphate groups. So this is a multi-step process. I do not expect you to really grasp and remember step by step by step by step, but I don't want to consolidate it and then you really don't have a clue of what's going on here. So we're going to highlight the main components, but also show you that it actually takes multiple steps to get there. So we start with glucose. Recognize that it's a 6-carbon molecule, so that's why we have those 6 orange dots there, or carbon atoms. There are no phosphate groups. The whole role of cell respiration is to get ATP out, but look at what we start with. We actually start with the input of ATP. So even before we get anything out of this process, we got to put energy in. So notice that it goes ATP, it exits as ADP. So there's 3 phosphates on this molecule, 2 phosphates on this molecule. So where'd that extra phosphate go? There it's accounted for. So notice I don't put names to these because I don't want to overwhelm you with those names because they're not that important. The later on in biology you go, or the higher levels you go, you might learn some of those names. So here we have, can you recognize that this should be a 6-carbon molecule with now 1 phosphate? So what will happen, another chemical reaction, no difference in the number of phosphates or carbon atoms. But then the next step, we introduce a second ATP molecule to produce a second ADP molecule. So now we have 2 phosphate groups on either end. So those 2 phosphates will go with their respective molecules. So there's a breakdown. Basically splits right in half to a 3-carbon molecule, each with 1 phosphate. This is a recognizable molecule. This is PGAL. We talked about that back in photosynthesis, where during the Calvin cycle, the PGAL was removed, and it took 2 of them to make glucose. So photosynthesis was more of like reverse all of these arrows, and that's how many steps it would have taken to actually get to glucose. But cell respiration is the reverse process. So PGAL, you have 2 of them. They both proceed through. The next step, here is a different type of molecule. You recognize from photosynthesis NADPH, and what this was an electron carrier. That was the electron carrier of photosynthesis. When it was carrying the electron, it turned into NADPH. This is the electron carrier molecule of cell respiration, NAD+. Notice that the big difference here is the drop of the letter of P, which I think is easier to recognize this because with photosynthesis, the carrier molecule has the name, the molecule in it is P. I don't think it stands for photosynthesis, but it's a good mnemonic device to recognize. So here's what happens. NAD+ goes in, it gets reduced, so therefore this thing is going to gain its electron. It got the electron from the PGAL, and it turns into the reduced form of NADH. When you take something away, you give the opportunity to add another bond because you broke one bond, so let's put another thing in its place. So here is the opportunity for a phosphate group. So PGAL had 1 phosphate attached to it with 3 carbons. Now the next step is 3 carbons with 2 phosphates because a phosphate group went into the process. Here, this molecule has a very high energy electron attached to it, which will once again play a role later on in this whole process. But for right now, we're going to account for it because it's a very important molecule. Side by side, the same process is happening over here because we have a second PGAL going through an identical process. So instead of just showing 1 and saying, let's imagine that it's happening twice, you're actually seeing it happen twice on this picture. So then we go through and we have the next step is actually where we produce an ATP molecule. And we're going to start counting how many things are made. And that's the big picture of this, is not knowing individual step by step when it happens, but we're basically trying to figure out how many of these very important energy molecules are produced. So here's our first production of ATP. So notice that the previous reactant had 2 phosphates, and ATP is made because a diphosphate was produced into an ATP, so we lost a phosphate. So that's why the next molecule only has 1 less than the previous reactant. So that happens over here as well. So another step, there's no production of any electron carrier molecules or ATP, so it's really somewhat an intermediate step. Happens again. So other things are happening here, but nothing with regards to carbons and phosphates. And then finally, the last step, we produce a second ATP. We have that 1 phosphate available, so that phosphate can then attach itself to the diphosphate to produce a second ATP from this PGAL original molecule. And what we're left with is a 3-carbon molecule with no phosphates. And this is a very important molecule for right now. It's the N molecule called pyruvic. Some text will put the form, it's more chemistry based. We're not really going to get into the dynamics of this. Sorry, I'm spelling it wrong. Actually, I'm not. So, they will call it pyruvic acid in some text. So they might say that glycolysis ends with 2 molecules, there's 1, there's 2, of pyruvate, or others might say that it ends with 2 molecules of pyruvic acid. Basically, to make it as simple as possible, they're somewhat interchangeable at the biology level we're looking at here. Some books might end all of their words with ATE, others will end it with ic acid. So there's a different form to it, I believe, but the whole idea here is don't get confused if one book uses one variation of the word versus the other. All right? So here, let's basically, at this point, I've circled some of the major things that came out. We're going to tally these things up. So in terms of ATP, we produced, so I'll put produced or product. We have 4 as a product. So that's 1, 2, 3, 4. All right, important molecule. In terms of the reactants, though, we required 2 of them. So sometimes they say, let's figure out the net gain. Net is after you take into account what has gone into it. So the net gain is only 2, because basically these 2 reactants have to kind of counterbalance the 4 products. So now we're at a net gain of 2. Other molecules that are important will be the NADH we're going to keep track of. So how many of those did we make? There's 1, there's 2, so we count for 2. And that accounts for everything. Things that such as ADP, we see that they're coming out, but my hope is by now, if you realize that ATP is a reactant here, then by default, ATP will transition into ADP. So the number of ATP reactants will be equivalent to the number of ADP products. Just like up here, when ATP is a product, what is needed as the reactant to make that? You need the ADP. So if I have 4 ATP products, I can guarantee I have 4 ADP reactants. So it's not really something you have to clog your memory with. These are the things that are probably the most important. And then the other molecules, by default, you can kind of figure out instead of memorizing double the amount of molecules. All right? So that's pretty important, where at this point, what we've done is we've taken some energy out of glucose. We haven't taken all of it. There's still a lot of energy left in these 2 molecules, but they're not as much as what we started with, because what happened is we took some of its energy with the NADHs. That electron that's on there is some of the energy that was stored. And then we also have ATP, which is obviously energy. So this molecule as a reactant must have more energy than its product here, because in the process, energy was released. So think of that as somewhat an exothermic reaction, because the amount of energy potential here is less in the product, because energy was lost right in between this reactant to that product. Okay? So that's glycolysis. We'll pick up the next video with the next step in the process.